Hello everyone, and welcome to episode 122. In today's show, we'll be continuing our exploration of the crustaceans with a mini-series on the Malacostracans. We'll begin by exploring the wide diversity of basal multicrustaceans, as well as simple Malacostracans, known more generally as the shrimp and the krill. These are quite small animals, but they are incredibly important. They're ubiquitous and cosmopolitan. They are perhaps the largest contributor to secondary productivity in all the oceans. They represent a colossal portion of marine organic carbon, and they're basically the backbone of the marine food web. Their ecological contributions drive much of the nutrient cycling and trophic activity of aquatic ecosystems. As you'll soon learn, many of the groups that we're going to look at today and many of the subjects that we're going to talk about in great detail are either extremely gross, extremely violent, an extreme life cycle, or extreme morphology, or some combination thereof. This episode is going to be a wild ride, as we're going to explore some of the craziest stuff in the animal kingdom. Now, in the previous episode, we explored the early evolution of the crustaceans. That episode began with a look at the evolutionary context in which the ancestral crustacean emerged, as well as an exploration of the Oligostracans, the oldest crustacean lineage with modern descendants that still live today. Now, the next major branch to diverge from the primitive crustacean stock, sometime in the mid-Cambrian, was the Multicrustacea. Now, the Multicrustacea is another basal crustacean branch, and it would actually end up producing the vast majority of all modern crustacean species. Nearly 80%, or 4 out of 5 crustacean species, are multicrustaceans. They fall into the Multicrustacea clade. And by the late Cambrian, Multicrustacea had split into 3 major classes. You had the Thecostracans, the Copepods, and the Malacostracans. Now, the Malacostracans are the largest class of crustaceans, with almost 40,000 species. We'll be exploring this vast Malacostracan biodiversity over the next few episodes, but today we're going to start with the simpler Thecostracans. Then we're going to move on to the Copepods, and then we're going to take a look at all the shrimp and the krill and everything else in the clade Malacostraca, with one exception. We're going to look at a lot of different groups today, but we're not going to look at the decapods. This particular Malacostracan order includes the lobsters and the crabs, and we'll be looking at these 2 groups in the next 2 episodes. For now, we're sticking with the simpler multicrustaceans and the smaller Malacostracans, like all the various shrimp and krill. Now that we have that housekeeping out of the way, let's get started. The story of the Multicrustacea and the shrimp and the krill begins in the early Cambrian, around 530 to 540 million years ago, shortly after the emergence of the multicrustacean superclass. The ancestral stock that would found the Multicrustacea superclass had a really fascinating morphology, which is all the more understandable in its evolutionary context. These ancestral crustaceans had descended from the basal arthropods, in a lineage that had positioned them as the sister clade to the myriapods. Now, the myriapod fossil record is poor and not well understood. There's a massive gap in the fossil record during the transitionary period from marine to terrestrial lifestyles, which is as disappointing as it is frustrating. But what we can piece together is that by the time the myriapods were on land, by the time they had adapted to these dry terrestrial soil-bound habitats, they had body forms quite similar to those that we see today. Long, narrow, almost strangely worm-like, but with many legs, long antennae, and wicked chemical defenses. Their aquatic ancestors were similarly long-bodied and many-legged, but had fewer segments and fewer pairs of appendages. They were smaller and stubbier, similar to the ancestral proto-crustaceans. These too were segmented and many-limbed, but were also somewhat short and stocky. Some early stem groups, like the Oligostraca and the Mysticocarida, evolved a more myriapod-like body form, with a longer, heavily segmented abdomen. Yet other groups, like the Branchiura fish parasites and the Ostracoda seed shrimp, had body forms that remained somewhat broad and consolidated, more compact, and less elongated, while still retaining the segmentation and many pairs of limbs. In addition, they evolved a two-plated carapace, whose armored sheets covered the sides of the head and often some of the limbs. Compared to the ancestral pancrustacean, the basal multicrustacean was similar in size and shape, as demonstrated by two distinct subclasses, the Copepoda and the Tantulocarida. Let's start with the Copepoda, as these represent a massive and cosmopolitan group of crustaceans that can be found basically everywhere there's water. In the ocean, on and within the seafloor, in freshwater lakes and rivers, and even in wet terrestrial environments like swamps, bogs, or water-saturated jungle soils. Now, copepod morphologies and lifestyles are as varied as the habitats they live in, but all of them are small, no more than 2 millimeters long, with a large head fused to the first 1 or 2 thorax segments, with no more than 5 additional thoracic segments, all equipped with hairy legs for swimming. They form either part of the plankton in the open ocean, or they form part of the soil microfauna communities on dry land or on the seafloor. Because they're so small, they can rely on diffusion for their metabolic needs. In other words, they don't need lungs or gills because oxygen simply diffuses into their tissues. They don't need a heart or blood vessels or any kind of dedicated circulatory system because nutrients effectively diffuse from their digestive system across all the tissues in their tiny bodies. Like the more basal Mystacocarida, the copepods are also cyclopean creatures, with a single, centrally placed eye, made red or black with photoreceptive pigments. There's even a widespread and abundant copepod genus called Cyclops, which typically lives in freshwater lakes. Although, like many myriapods and basal crustaceans, those copepods that live in the seafloor sediments, or in the cold, dark water of a cave, typically evolve to lose their sense of vision, and even their entire eyeball, or their eye structure. Also like the Mystacocarids, the copepods are often hairy, or bristly, and equipped with long and sensitive antennae. To scientists' surprise, the copepods are particularly sensitive and quickly responsive to environmental disturbances like an approaching predator. This is due to the neurons, the neural tissue in the antenna and the rest of the body, which is protected with a kind of myelination, which insulates the neuron and increases the speed by which action potentials, or chemical sensory information, travels down the axon. This is a particularly unique crustacean development, similar to vertebrate neural myelination. This advantage, however, only exists in some copepod groups, but not all of them. Their movement patterns, how they move their body to get through the water, depends on their energy expenditure and their method of locomotion. Some of them may take brief rest periods, wherein they're totally immobile, while others are constantly moving. Some may start and stop moving, regularly or irregularly, while others are more fluid and continuous. As they zip about, avoiding predators and taking advantage of small currents in the water, they're feeding on a massive amount of algae and phytoplankton, so much so that these copepods can actually help to contain algal blooms. In this way, many of the copepods are primary consumers. They consume the biomass produced autotrophically through photosynthetic sugar fixation into heterotrophic carbon in the body of an animal, or in their body. Also, the larger copepods are known to eat smaller copepods. About half of the copepods are parasitic to some degree, spending part of their larval stages in the body of a host animal, like a sponge, or some coral, or a slug, or a sea snail, or even by parasitizing vertebrates, like sharks, seals, and smaller fish. There's a very fascinating phenomenon at work here. These parasitic copepods have experienced an evolutionary explosion as clade after clade rapidly adapts to a host. As a result, the parasitic copepods are extremely diverse in shape and form, and even in the particular regions of the host body that they occupy and how they parasitize it. When it comes to mating in copepods, the process is pretty straightforward. Pheromones are used to attract mates to each other. Then the male will produce a spermatophore, and he'll use his many legs to shuttle it from his genitals across his body to the female's genital pore. After the eggs have been fertilized, they'll spend just a short time developing in the female's body before she secretes them into the external environment. Now, some copepod species will just leave their eggs floating in the water column, their extremely small size a kind of defense of its own against predators. In other species, the female will create a small, filmy sac or bag in which the eggs are stuffed. The bag is then glued onto her carapace with some sticky secretion, and the eggs will trail along with the mother until they grow large and hatch. The newborn larvae are literally just heads and tails. There's no thorax, there's no abdomen yet. It takes 5 or 6 molting events to achieve a larval stage, and another 5 or 6 molting events to reach maturity. This may sound like a lot, but it depends. It varies a lot based on the species and their nutritional income. In the best case scenario, where the specimen belongs to a fast-growing species and lives in an area with abundant food resources, it may reach maturity in just a week. In other cases, with a specimen of slower-growing species suffering from poor food availability, they may take a year or more to molt those 10, 11, 12 times before reaching adulthood. Closely related to the copepods are the Tantulocarids, a relatively small subclass with a mere 33 primitive species. The creatures themselves are kind of like copepods, but particularly small, less than 1/3 of a millimeter long, with the smallest among them, Tantulocarid diateri, being the world's smallest arthropod. These are 85 micrometers long, which is less than 1/10 of a millimeter. They're also quite primitive, with a reduced thorax and abdomen, and a fully parasitic lifestyle. Tantulocarids seem to have a preference for their fellow crustaceans. They parasitize a broad variety of smaller crustaceans like ostracod seed shrimp, tineids, amphipods, copepods, and isopods. They'll use hooks or suction cups to attach to the softer tissues on the host, like around the eyes and the mouth, or around the joints along the limbs, or the softer ventral surface of the body. Then they'll use sharper mouth parts to make a tiny slice in these delicate, heavily vascularized tissues. The cut is very small, no larger than the diameter of a bacterial cell. A primitive cell mass is then pressed into the cut, which grows like a plant's roots or rhizomes into any of the host's soft innards that happen to be nearby. This parasitic growth extracts nutrients from the host, sort of like a saprophytic fungal infection. The nutrients are absorbed by the Tantulocarid, which uses them to fuel more growth of this parasitic, fungal or root-like mass that extends deeper into the host's organs and tissues. This is so gnarly, because it's kind of like an animal's attempt at a fungal growth form and lifestyle. And as a weird twist, the whole dynamic kind of runs in the opposite direction, where the fungus invades a host with mycelia first, and then uses stolen nutrients to form fruiting bodies that protrude outside the host's body. The Tantulocarids attach themselves to the outside of the host's body first, and then surgically penetrate and invade the host with a squirming mass of absorptive tendrils. Now, these Tantulocarids that are mounting themselves onto a host, slicing it open, and then growing this weird parasitic mass into its soft tissues, these kinds of Tantulocarids are parthenogenic females. They produce a single clutch of eggs with their own genome, and then the clonal egg sac sloughs off into the water. Sadly, the detachment of the egg sac causes grievous trauma to the mother, who dies shortly after. Now this is where things get really weird. Some of the offspring will grow from a tiny embryo to the complete adult body form, all inside the egg. When they eventually hatch, they're tiny but fully mature adults. This accelerated development process within the egg is believed to be caused by evolutionary forces discouraging a wide dispersal of eggs and the birth of underdeveloped larvae, as their adaptations for a parasitic lifestyle have driven them to immediately seek a host after they're born, and they can only do this with a fully developed adult body form. You might be wondering why, a few moments ago, I said that only some of the offspring grow to adulthood inside the egg. You might be wondering, why not all of them? Well, that's a great question, and it has to do with the evolutionary context of the Tantulocarids. These parasitic females are parthenogenetic, so they're operating on the basis of clonal reproduction, which inherently has limited genetic variation. Clonal reproduction, or asexual reproduction, runs the risk of accumulating defects in the genome, which can eventually cause a lineage to weaken and succumb to a disease or an environmental change. Genetically, too many flaws and problems and recessive alleles get built up, and they just get burned out. Naturally, there's an evolutionary pressure for asexual lineages to somehow find a way to avoid this terminal spiral of genetic degradation. In bacteria, for example, and many species of microbe that reproduce through mitosis, they can enrich and diversify their genomes through horizontal gene transfer and other lateral techniques like that. They don't necessarily rely on the vertical transmission of genetic diversity. More relevant to today's topic, in the Tantulocarids, they've developed a reproductive strategy that combines asexual and sexual replication. The parasitic females create a clutch of eggs through parthenogenetic reproduction. However, in the batch of eggs, only some of the eggs are females that are genetically programmed to become parthenogenetic parasites, just like their mother. But other eggs in the batch, which can house male or female embryos, these are genetically reprogrammed to avoid the parasitic life strategy. As they do not become parasites, they're not exposed to the same evolutionary pressure to develop rapidly into the adult form inside the egg and find a host as quickly as possible after hatching. This is what we see in those particular females that are genetically programmed to become obligate parasites and reproduce asexually. But in all the other eggs, in the males and females that are destined to be free-living, sexually reproducing adults, well, they mature a little bit slower in the egg. They're much more like other crustaceans in that sense. They hatch not as fully grown adults, but as nauplii, an intermediate life stage, sort of like an immediately post-larval stage that gradually develops into the adult form. These offspring that hatch as nauplii will scavenge about, finding food for themselves and growing a little bit larger, and eventually developing into the adult form, where they find mates and sexually reproduce. This is a really interesting evolutionary strategy. The Tantulocarids have evolved two distinct but integrated lifestyles, where one supports the other. The parasitic Tantulocarids are reliable and can asexually produce many offspring by tapping the nutrients in a larger host. While the free-living, sexually reproducing groups, they may produce smaller clutches of eggs, but these have enriched genetic diversity and allele heterozygosity. These benefits are then passed on to their offspring, which can be destined for either parasitism or autonomy. In this way, the sexually reproducing groups create the genetic diversity in the shared gene pool that prevents the asexual lineages from spiraling into terminal genetic degradation. It's an ideal strategy to sustain the overall population, when part of the population is locked in a really extreme mode of life. The obligate parasite niche can be extremely difficult for a species to escape from, especially if it reproduces asexually. If the host goes extinct, the parasite goes extinct too, unless it can quickly adapt to a new host, or a non-parasitic lifestyle. And that can be difficult, or even impossible, depending on how specialized the parasite is. And reproducing asexually will make survival in this condition even harder, because it slows down the evolution of new and beneficial traits that would help it achieve a new lifestyle, and survive, and not go down with the ship, so to speak. There's one more detail I wanted to include before we move on from the Tantulocarids. In this batch of eggs, even those produced by the parthenogenetic females, there's going to be a mixture of male and female offspring. The females, both those that develop quickly to become parasites and those that develop a little more slowly to become autonomous, they exhibit a normal growth pattern in the egg. They grow like any other egg-bound embryo, which kind of sounds vague and unhelpful when I say it out loud, but perhaps it'll make more sense when I describe male development. The males have a distinctly not normal growth pattern. In the male eggs, an embryonic mass develops into the beginnings of a larval body, with a weakly defined head, thorax, and abdomen. Curiously, this larval body is neither male nor female, and after a short while, the thorax begins to change. The tissue begins to take on a new shape, and a new, unusual mass grows out of it. This new mass protruding from the middle of this weird asexual larval embryo is, in fact, the male's actual body. And that initial larval body, the one that grew first and is sexually undifferentiated, that basically gets reduced to being a de facto umbilical cord. To illustrate this really viscerally with a human analogy, this would kind of be like if a human embryo grew a teratoma early in the development process, except the teratoma grew faster than the embryo and stole so many of its nutrients that it effectively turned the embryo into an extension of the umbilical cord for its own growth. So whatever gets birthed is in fact the teratoma and not the original embryo. That's what the embryonic development of the male Tantulocarid is like. Now, closely related to the Tantulocarids are the Thecostracans. They're more commonly known as barnacles, after the largest Thecostracan subclass, Cirripedia. There's two other subclasses, although they're really small and not very biodiverse. These are the Ascothoracida, which are small parasites that infect sea anemones, starfish, sea slugs, and the like, and the Phacidotecta, which are also suspected of being ectoparasites like the Tantulocarids and the Ascothoracida. But no one has actually seen an adult Phacidotectan, so no one knows for sure. Anyway, the true barnacles are shallow water crustaceans that anchor themselves to a substrate and stay there, immobile, lazily filter feeding for basically their whole lives. This process begins with the one-eyed larva, which, like many other primitive crustaceans, is born as just a head and a tail segment, which grows a thorax and an abdomen later through successive molting events. After 5 or 6 molts, the newborn larva becomes a cyprid larva, which is kind of like a crustacean analog to a teenager. This is a period of exploration in the barnacle's life. The cyprid larva roams around its shallow water environment, using its antennules, its smaller secondary pair of antennae, to feel around and to seek out a suitable place to anchor down. This is really the only time in its life that it's going to be able to move around under its own power and its own will, its own direction. But unfortunately, it has to be quick, because it really doesn't have that much energy to swim through the water and look around for a place to live. As time goes on, the larva will become less and less picky, because it has to find somewhere before it exhausts itself. Once a good location has been identified, the cyprid larva will attach itself head first, using a pair of glands at the base of its antennules. These glands secrete highly adhesive glycoproteins that create the permanent bond between the barnacle and its substrate. Depending on the species, the barnacle will either glue itself directly to the substrate, or it'll have a stalk connecting its main body mass to rocks on the ground, or a coral reef, or whatever the substrate happens to be. Once the cyprid larva has secured itself, it'll begin maturing into adulthood by secreting a calcareous shell matrix. This calcareous shell matrix creates 6 curved plates that surround the body like a big pot or a kettle. Its legs can stick out of the top, out of the open end of this 6-plated bell or pot-like carapace, where they wave around and capture food particles in their little hairs. Their anal and genital orifices also protrude out of this back end, or top-side opening in the shell. In some clades, the barnacles have evolved additional carapace plates to cover this opening, offering a little bit more protection against predators and diseases and pathogens like fungus. Generally speaking, these barnacles are really interesting from an ecological perspective. They mostly live in shallow water environments like sunlit reefs and intertidal zones. Within these habitats, various species of barnacle have adapted to extremely specific sub-habitats. For example, some species might only be found in the shaded southern side of a tide pool. Others may only be found at the edge of low tide, so that they're always submerged, while others still may live closer to the shore, on rocks exposed to air. By studying the particular species composition of a fossilized barnacle community, scientists can make all kinds of estimates, from the water level in the habitat to the water's pH and salinity. And then you have barnacle reproduction, which is also really interesting and super weird because they had to evolve solutions to problems that arose with their permanently sedentary lifestyle. Because adult barnacles are totally immobile, they can't move at all, so they can't search around for mates or do a mating ritual with them or anything like that. They had to devise alternative strategies. Spraying sperm into the water in the hopes that the cloud of gametes will come into contact with a receptive female is one strategy that males of some species use, although it's a pretty inefficient process that doesn't give males or females much ability to choose their mates. So the major disadvantages are that it's random and inefficient. To avoid these problems, the barnacles have evolved several other reproductive strategies. For example, hermaphrodism is very common among barnacles, although, curiously, self-fertilization isn't very common. Perhaps because the barnacles have evolved ways to avoid self-fertilization, to avoid inbreeding depression and other issues with poor genetic diversity. Instead, it seems like the high frequency of hermaphrodism among the barnacles simply makes it easier for dyads or mating couples to meet and do the deed. For example, if there's just males and females, then any given individual in an area could either be a suitable mate or not. But if the species is all hermaphrodites, then any given individual could be a suitable mate for any other. Now, of course, there's some species that are gonochoric, where they just have a male and female sex with no hermaphrodites, and they don't spray sperm into the water either. Instead, in these particular gonochoric barnacles, their reproductive evolution has crafted enormously long penises, up to 8 times their body length. These exceptionally long penises can protrude from the shell and then feel around the local environment to find the shell opening and the genital pores of a receptive female. Now, this method is much more targeted and much more efficient than just spraying sperm out into the water, and it also has the benefit of avoiding any genetic risks from hermaphrodites' self-fertilization. Now, what I've described here, from the larva anchoring to a substrate, to filter feeding, to these various forms of reproduction, all of this applies to most barnacles, and to most Thecostracans. However, there's one group of cirripedian barnacles that's quite unusual. This is the superorder Rhizocephala, and these rhizocephalans are incredibly weird creatures. they have massively reduced and simplified bodies due to their entirely parasitic lifestyle. The adults don't have any limbs, which is uncommon but not altogether too strange. But they also don't have any segmentation, which is kind of strange for an arthropod. And they don't have any internal organs. And, okay, that's definitely weird. The females, for example, just have gonads for producing eggs. That's it. That's the only internal organ structure they really have. They're intuitively very tiny, so nutrients, gases, and water can all be absorbed through their bodies by simple diffusion. Beyond this, they have small tracts of borderline vestigial muscle tissue and a much reduced nervous system. Now here's where things get really crazy. Like the other barnacle larvae, and like the Tantulocarids, the Rhizocephala larva is free-living, free-swimming, and can search around for a suitable host. When a host is located, the larva approaches and anchors its head to the host's epidermal tissue. But instead of then secreting a calcareous shell like other barnacles, the Rhizocephala larva will produce a vermigon, a specialized mass of cells that gets injected through a syringe-like structure on its head into the hemolymph and soft internal tissues of the host. In this way, they use a strategy much more akin to the Tantulocarids than other barnacles. Anyway, once they've made it inside the host's body, the vermigon cell mass will proliferate, growing kind of like a fungal mycelia. The vermigon forms rhizome or hyphae-like structures, thin filaments of cells that spread through the host's body. Speaking of the host organism, the Rhizocephala only parasitize decapod crustaceans, like crabs and lobsters, as well as a few clades of shrimp and other barnacles. Because all of these host animals are arthropods with exoskeletons, the invading tendrils of the vermigon creep down the chitinous corridors of its many limbs to feed on the soft muscle tissue and the hemolymph soup that it bathes in. The main priority of the vermigon growth is not the limbs, but the host's digestive system, where it can find and tap into the greatest concentration of nutrients in the host's body. Now, after this infection has developed substantially, the host will show relatively few outward signs of this parasitic infection, unless you know where to look and what to look for. The only externally visible evidence of a Rhizocephala infection is a slimy, smooth, fleshy, and bulbous organ called an externa, which protrudes from the belly of the host. The externa is typically a nauseating yellow to a milky off-white color, and it's basically the Rhizocephalan's reproductive organ, with a genital orifice with a single ovary, and typically 2 passageways leading to sperm receptacles, or gamete storage chambers. So what, you might be wondering, is the male doing this whole time? If the male has no external organ analogous to the female, how do they reproduce? There's a lot of variety when you look at individual genera and species, but the general idea seems to be that a young male, a larva, swims into the mature female's externa, down the passageways and into the sperm receptacle chambers. Once it arrives here, the male larva will extract its own cellular mass from within its own primitive exoskeleton. To put this another way, to the best of my understanding, the male undergoes some process by which the internal cellular mass, the little bit of soft tissue within its larval exoskeleton, is extruded through an organ that's analogous to the one females use to extrude the vermigon and initiate a parasitic infection. The male larva, attracted to the external passageways with pheromones and embedded deep into the cell receptacle chamber, will then deposit his cellular mass, the entire soft tissue mass of his whole body, into the female's externa, where his cells can be transferred to the ovary. The male's cell mass undergoes a transition, kind of like a metamorphosis, into a new life stage, except here, its already degenerated body is further reduced into a small rag of cells that just produce sperm. If you thought there wasn't a worse parasite than your best friend's loser boyfriend, I might have been able to change your mind by introducing you to the anglerfish, of which the male will fuse himself via his lips to the underside of the female and become a glorified testicle or a scrotum sac providing sperm to the female on demand. Well, here I think we've found a case of parasitism that's even worse. This life stage is remarkable. The male Rhizocephalan is a hyperparasite on the female to which it's mating. The male survives entirely by diffusion of nutrients from the female, and the male's form is basically reduced to the absolute bare minimum it can possibly be while still technically being a male animal. He's basically been reduced to a cluster of cells, more like a film, like an inverse nicotine patch, that can turn nutrients given from the female into sperm, which the female then uses at her discretion for fertilizing her eggs. The creepy factor doesn't end there. The host species usually produces an egg sac of its own, but when it's infected with a Rhizocephalan parasite, its egg sac is replaced by the parasite's externa. I don't think it's understood just how much influence the vermigon structure has on the behavior of the host, but it has been observed that the host will protect and brood the externa as if it was its own egg sac. Even male hosts, like male crabs or male lobsters, who would never have had an egg sac of their own in the first place, have been observed exhibiting brooding behavior and protecting the parasite's egg sac as if it was its own. Okay, that was thoroughly disgusting and awesome at every step. Let's take a pause for a moment to take stock of where we are. So far, we've explored the anatomy and ecology of many of the simple, primitive multicrustaceans. We saw that some of them were part of the zooplankton, and thus a critical part of the marine food chain. But many of them were also parasites that invaded fish, soft-bodied invertebrates, and other crustaceans. Well, now, at this point, we're going to transition to talk about the Malacostracans. The two most basal Malacostracan lineages are the Phylocaridans and the Hoplocaridans. At one point in the ancient past, both of them were much larger and much more diverse, but they've since been whittled down by evolutionary forces, and today each lineage only contains a single extant order. The Phylocaridan lineage emerged in the early Cambrian and represents perhaps the most ancient and primitive Malacostracans. The only surviving order is the Leptostracans, which are small, shrimp-like creatures, about half an inch long or smaller, with a round, thumb-shaped carapace that covers their head and most of their thorax. Where classic shrimp have a tail that curls down under the belly, the Leptostracan tail instead bends up and extends straight out. These creatures live all over the oceans, predominantly in the intertidal zones and other shallow water habitats. They form an invaluable part of the food webs here, where they filter feed on small food particles in the water and are preyed upon by all manner of larger predators. Like many of these crustaceans, they're an important intermediary link in the food web, facilitating the flow of nutrients from the scale of the microscopic to the macroscopic, with larger animals preying on each other and shuttling nutrients through the trophic chain. The other basal Malacostracan lineage is the Hoplocaridans, which emerged 340 million years ago and whose only extant order is the Stomatopoda, which emerged 193 million years ago. The Stomatopoda are more commonly known as the Mantis shrimp, which are very colorful and charismatic crustaceans. In contrast to virtually everything I've talked about so far today, the Mantis shrimp are pretty large and beefy, with most individuals growing between 3 to 5 inches long, with some species growing over a foot long. They dwell on the seafloor, where they scurry around in the shadows trying to find food or shelter. Some of them prefer to live in rock formations or coral reefs, or other places with hard rocky substrates. while other mantis shrimp prefer areas with softer substrates like sediments and sands, where they can burrow in and build their own shelter. The mantis shrimp spend most of their time here in these protected home burrows, only venturing out either to scavenge or to hunt, or to find a new home to move into. When food has been captured, they take it back to their protected burrow to eat. When a predator attacks them, they'll retreat back to the burrow. When a mate has been found, they'll take them back to their burrow. On that note, depending on the species, the mantis shrimp are either monogamous for life, or they only interact with the opposite sex when they're seeking a mate to reproduce. In the monogamous mantis shrimp, there's also some variety in their relationships. Some species are co-equal, with both parents hunting and brooding the eggs. Some species go so far as to have the female birth 2 clusters of eggs, share one of them with the male so both of them have eggs to brood over, and then they go about their daily business, like finding food. Conversely, the other monogamous mantis shrimp couples are more specialized, wherein the female performs most or all of the egg care and the brooding, while the male does most or all of the hunting for them. The male goes out and captures food and brings it back to feed everybody. Now, there's a lot more that I can say about these animals. They are unique and super fascinating predators. They have numerous traits that are extreme or unusual, and there are several really interesting patterns in their distribution and behavior. To keep it brief, let's take a look at 2 of their more extraordinary features: their eyes and their claws. To begin, the eyes of a mantis shrimp are unbelievably sophisticated. This random, rather primitive clade of Malacostracan possesses some of the most overdeveloped visual hardware in the world. The complexity of their eyes is unmatched, and the implications for their visual abilities and their intelligence are staggering. To give you a sense of perspective, no pun intended, the mantis shrimp have 4 to 5 times as many different types of photoreceptor cells as humans do, and many of them detect ultraviolet light. These photoreceptor cells are grouped in clusters in pixel-like ommatidia structures, and each eye is made up of many thousands of them. The eye itself is organized into 3 sections. There's 2 semicircle regions separated by a central strip, or a belt-like region. The ommatidia are geometrically arranged in complex patterns, with specific photoreceptor types in specific areas. Each section of the eye can see on its own, meaning that each eyeball technically contains 3 miniature eye structures. This gives each eye trinocular vision, which means depth perception, so as each stalked eye is swiveling around independently, it can still judge vectors and distance and approach speed of other creatures in the environment. They have powerful visual abilities, depending on the species. For example, there are some species that exhibit some degree of spectral tuning, wherein they can optimize the sensitivity of photoreceptors that respond to light with long wavelengths. This long wavelength light is like oranges, reds, and browns, which quickly fades to black in the light of an underwater environment. By using spectral tuning, the mantis shrimp can enhance these colors and see prey animals that have evolved red shells specifically as a means to melt into the aquatic darkness. In other words, some prey species have evolved to have red shells, because deeper in the water where the light is pretty dim, having a red shell makes you basically look black or invisible. It's like an invisibility cloak. And this spectral tuning ability that the mantis shrimp use to enhance these colors allows them to see the red shell instead of the black blob or the invisible shape. It's basically an evolved countermeasure to defensive cloaking. Furthermore, several species have been known to detect circularly polarized light, which is something that no other animal can do. In this context, the purple spot mantis shrimp, Gonodactylus smithii, is believed to have the best visual system in the world, able to detect essentially all types of polarized light. This remarkable feat is achieved by rotating the eyeball in the socket, not side to side like it's looking around, but rotating along a central axis, like a gear cog or a steering wheel, inside the socket. Even the stalks that the eyes sit on are unusually flexible and dexterous compared to virtually any other animal that also uses eye stalks. By using their sensitivity to polarized light and ultraviolet light, in combination with their ability to fluoresce, allows them to communicate non-verbally with light patterns that other animals can't detect. They can seek out mates without alerting predators. They can more easily detect predators and prey alike in the dark seafloor environment, and they can respond extremely quickly to movement close to them, like an attacking predator, a competing mantis shrimp looking for a fight, or a prey animal attempting to escape. Their claw-eye coordination is exceptional, and this is just part of the reason why they're dangerous and hyper-violent predators. Mantis shrimp have claws that can be loosely organized into two groups, spearers and smashers. The spearer mantis shrimp have spear-like claws, with long, thin spikes with barbed tips, which they use to stab their prey. The smasher mantis shrimp have club-like claws, which are used quite effectively to beat up, smash, and pulverize their target. These brutish claws aren't just simple chitinous clubs. They also contain a small mini-spear structure, like a thumb, as well as a bladed edge along the inner margin, both of which are used to hunt prey and to fight off other mantis shrimp. There are smaller groups of lesser-known mantis shrimp who have axe or hatchet-like claws, and a purported missing link type group with spiked smashers, or mace-clawed mantis shrimp with elements of both club and spear claws. Perhaps the most dangerous aspect of the mantis shrimp attack is not their claw-eye coordination, and not the brutal implements that they use to shred and kill their target, but rather the speed at which they strike. This really doesn't seem like it should be physically possible, but mantis shrimp can strike out with their claws at an acceleration of over 10,000 Gs to reach almost instantaneous speeds of 23 meters per second. This unbelievable strike speed creates friction, a lot of friction, which rapidly heats the water so much that it creates cavitation bubbles. This is basically an explosion underwater that creates a brief vacuum space that then collapses in on itself. So you have the initial explosion, and then you have this immense force of the water collapsing in on the open space. If you're some poor little reef fish swimming around looking for some algae to innocently nibble at, an unseen mantis shrimp can pop out of its burrow, hit you with a club or a spear claw at 1,500 newtons, immediately turn your insides to pulp, and then you get hit with the force of the collapsing cavitation bubble. Even a near miss with the claw can still be fatal because of this collapsing cavitation bubble shockwave. Because of their insane speed and force, mantis shrimp can hunt and kill animals much larger than themselves, like fish or cephalopods, even armored or shelled animals like snails, bivalves, and crabs. The mantis shrimp can handle these larger prey animals by punching or snapping at their legs, which can maim or cripple them. Or they can punch or snap at the larger animal's face and destroy part of their head, turning their eyes to pulp or just caving in the entire head structure. These stomatopod mantis shrimp are just crazy animals in so many ways, which is even crazier when you consider that they're the most basal Malacostracans. When they branched off way back in the Cambrian, the stem group that they left behind would become the U. Malacostraca, and it would include all the rest of the Malacostracans. Within this Malacostraca subclass, the very first big division would occur about 420 million years ago, and it produced a lineage known as Decapoda, or the decapods, for their 10 legs. This is a titanic order that contains the bulk of the biodiversity among Malacostracans, and, well, basically all crustaceans. Pretty much all the crabs and lobsters and crawdads that you've ever heard of are all decapods, in the order Decapoda. And I actually don't want to get into them too much today, because the next 2 episodes will be all about them, all about the lobsters and crawdads, and then the next one all about the crabs. So for the rest of this episode, we're going to look at the anatomies and life cycles of the remaining Malacostracans that emerged after the decapods. These remaining Malacostracan crustaceans can be broadly divided into 3 groups. First, you have the order Euphausiacea, which includes all the krill, as well as 2 superorders, Paracarida and Syncarida. Now all of them look like tiny shrimp of some form or another, and most of them are part of the zooplankton, which is a larger community of microorganisms that sustains the marine food web. The Syncarida superorder was the next lineage to emerge after the decapods, sometime between 410 to 380 million years ago. Syncarida is relatively small and poorly understood, containing only 2 orders, the Anaspidacea and the Bathanellacea. Both orders contain a handful of tiny krill-like species, found either in caves and freshwater habitats, or groundwater habitats, respectively. The Paracarida superorder emerged next, around 365 million years ago, and it would be much more successful than the older Syncarida superorder. Paracarida would produce a wide range of descendant lineages, although the exact number is debated. The molecular data here has been challenging to decipher, and so the precise phylogeny of the Paracarids is unknown. However, a trait that unites them all is the marsupium, which is a brood pouch on their bellies where they store the eggs as they develop and hatch. There are several other traits that most of them have, although this varies from order to order. For example, the carapace, the armor plate that protects the head and the thorax, is reduced or even absent in some groups, but not in others. Consider the Lophogastrida, which are deep-water crustaceans with an overdeveloped carapace. It extends back beyond their head like the frill of a triceratops. Similarly, the Cumacea hooded shrimp have enlarged head regions with a similarly bulbous carapace. In contrast, the tiny Myctacea and the varied Amphipoda don't have a carapace at all. The Myctacea are small, weak, stubby-limbed, and blind, like the Bathanellacean syncarids, whereas the Amphipoda are much more diverse and interesting, but they just don't have a carapace. The Paracaridans also live in a wide variety of habitats. The Lophogastrida I mentioned, the ones with the big frill-like carapace, live in the deep sea. The Speleogryphacea are only found in caves, and the Thermosbenacea only live in hot springs. Other groups show a bit more diversity in the places they live. Both the Cumacea and the Tenatacea live in the mud and the sand of the seafloor, but this includes the seafloor of the deep ocean up to the intertidal zone and even estuaries and river deltas. And then you have groups like the Masaida, which are found in aquatic habitats all over the planet, including the deep ocean, caves, estuaries, rivers, lakes, and the intertidal zone. The 2 largest and most diverse Paracaridan orders are the Isopoda and the Amphipoda, which have around 10,000 species each. Their member species can be found in virtually all habitats, from marine and freshwater ecosystems to terrestrial ecosystems. Now, most amphipods are small in size, but bulky in shape. They can be measured in single-digit millimeters, sometimes even less than 1 millimeter. But larger individuals have been found at extreme depths, like the several-foot-long Alicella gigantea specimens. The isopoda are similarly bulky, but can grow to much larger sizes, with the largest species being the giant deep-sea Bathynomus isopods. The amphipods include both ground-dwelling and free-swimming species, which has no doubt contributed to their widespread distribution. In contrast, the isopods are only ground-dwelling, so they can't easily move to new territories, because they have to crawl around to get there. The amphipoda are mostly detrivores and scavengers. They eat dead stuff, like marine snow, the dissolving corpses of whales that sink to the bottom, and even their own exoskeletons after they molt and shed it off. In terms of their diet and lifestyle, the isopoda are way more diverse. Not only are there isopod detrivores and scavengers, there's also isopod filter feeders, herbivorous browsers that pick at algae and kelp, active predatory carnivorous isopods, and a wide variety of parasites that infect fish and other crustaceans. One of the more infamous examples of isopod parasitism is the Cymathoa exigua, which infects the spotted rose snapper Luteolus guttatus. The Cymathoa exigua will then damage the fish's tongue, causing it to wither and rot and fall off. Then the isopod will snuggle itself into the fish's mouth and functionally replace the tongue, helping the fish break apart its own food. Of course, it's a parasite, so it takes some of that food for itself. The whole arrangement is freakishly reminiscent of the Alien from James Cameron's 1979 film Alien. Curiously, these two orders, Isopoda and Amphipoda, also include some of the only crustaceans with fully terrestrial lifestyles. From within Amphipoda, there's a family called Talatridae, or the landhoppers and sandhoppers. These are little pill-shaped crustaceans that hang out near the shore, where they scurry around looking for rotting seaweed. The European landhopper, Talatrus saltator, buries itself in the sand or dirt near the coast, and comes out when the tide recedes to scavenge wet, limpy strands of seaweed from the intertidal zone. From within the Isopoda, there's the suborder Onycidea, which includes the woodlice. Depending on the species, they may live near the coast and be only partially terrestrial, or they may be fully terrestrial. These land-based isopods are really important detrivores, responsible for macro-scale breakdown of wood, bark, leaf litter, and other vegetable detritus. Their activity degrades this detritus such that other organisms, like microbes or protozoans, can more effectively break them down at a microscopic chemical level. In that sense, they inhabit a valuable ecological niche alongside the millipedes. And lastly, there's the order Euphausiacea, also known as the krill. These small, shrimp-like creatures have a cluster of long, thin legs sprouting out from the fused cephalothorax, and a long segmented abdomen equipped with swimming legs and a tail fan at the terminal segment. Their thoracic legs, the long and thin ones, are equipped with dozens of very thin fibers or hairs. These are arranged in parallel, like teeth on a comb, and the krill use these to catch food particles in the water. The long legs bring the food particle to the mouth, and the food is ingested. Most krill eat this way, making them omnivorous filter feeders. Common food items include phytoplankton, like diatoms, dinoflagellates, and other types of algae. As a quick aside, some of these dinoflagellates produce a protein called luciferin. Many different organisms express luciferin, from fireflies to snails to ostracods, and all manner of microbes. Luciferin is a molecule that can react with the luciferase enzyme in an aerobic environment to produce light. It's suspected that the krill don't actually produce their own luciferin, but instead ingest dinoflagellates that are rich in luciferin, and this is only slightly altered during digestion and absorption as it gets integrated into the krill's tissues. The krill have evolved to concentrate these fluorescent molecules in photophores, which are complex eyeball-like structures with rotational muscles and lenses and everything, except they're not used for sight, but rather for displaying this visual signal, this fluorescence. It's believed that the krill use these movable, rotatable photophores to signal to mates and communicate environmental information to other krill. Curiously, krill species in northern latitudes are more active hunters with a more carnivorous lifestyle. They hunt zooplankton, including many species of Copepod. This carnivorous preference for active hunting is also seen in a few krill species who have shorter arm hairs and are thus less effective filter feeders. Individual krill are quite small, usually just 1 to 2 centimeters long, with only a handful of species growing any larger. Most of these larger krill, which can reach 5, 10, even 15 centimeters long, usually live in the deep ocean, where there's relatively little light and the pressure is significantly higher. Despite the individual krill being tiny, they are prolific reproducers that form enormous swarm-like populations that saturate the world's oceans, usually between depths of 300 and 600 meters. Although there are species that live near the surface, and those that live as deep as 4,000 meters or 4 kilometers below the water's surface. All of this krill, collectively, forms hundreds of millions of tons, maybe even billions of tons of biomass. And all of this biomass feeds innumerable small to medium-sized predators, from fish that can outpace them, seabirds that attack them from the surface, squid and octopus that ascend from the ocean depths, as well as penguins, seals, and most famously, the great baleen whales, who open their enormous mouths to gulp in hundreds of gallons of water, enough to fill a small swimming pool. And then, with their jaws closed and baleen in place, the whale pushes water out of its mouth with its 6-meter-long tongue. All of the smaller stuff in that water, like zooplankton, like krill, gets caught in the baleen. The whale then pulverizes all of this biomass with its tongue, turning it into a mushed-up orangey pulp, which it then swallows and digests. These feeding gulps can extract a thick, heavy, protein-rich layer of pulverized krill, especially when the whale is feeding near a swarming event. Krill can exhibit swarming behavior, where thousands of them rush around like crazy in a dense area. They do this for defensive purposes, to make it harder for predators to pick out individual krill. When krill swarm like this, the populations can become extremely dense, with anywhere from 10,000 to upwards of 50 or even 60,000 individuals per cubic meter. Such dense masses of krill can attract all manner of seabirds, fish, and marine mammals, leading to an aggressive feeding frenzy. These are some of the ways that the krill provide an extremely important ecological service. They're one of those key species that consumes single-celled organisms and then concentrates those nutrients and chemical energy into a larger body, into a larger organism that can be preyed upon and thus transfer nutrients to even larger predators higher up the food web. They are thus a critical bridge for the transfer of nutrients from the microscopic realm to the macroscopic food web. And their sheer quantity, the raw biomass, is a fundamental part of their ecological value. The krill are a food source for virtually the entire ocean. The number of aquatic species who feed on them, whose populations are utterly dependent on them, exist across the world and are far too numerous to count. In addition to being a bulk food source for the world's oceans, all of these krill, collectively, are massively influential machines of nutrient cycling. Populations and swarms of krill are followed by trails of debris including respiratory gases, molted exoskeletons, and excreted waste. Their gills release ammonia and phosphate. Their rotting, dissolving exoskeletons release calcium, fluoride, phosphorus, and small bits of organic carbon debris. Their urine is rich in ammonia. Their fecal pellets are rich in iron, and both are rich in dissolved organic carbon, nitrogen, and phosphorus. The swimming motion of their legs stirs these nutrients into the seawater, and so everywhere they go, they produce vast quantities of these substances and mix them in. This, in turn, feeds the microbial communities that the krill themselves rely on for food. It's a beautiful cycle. You can see how so many of these crustaceans, including many Malacostracans, are invaluable components of the food web and various chemical cycles. They are invaluable parts of the global ecology and the dynamism and resilience of biological ecosystems. But many of them are threatened by climate change, and their diminishment or extinction will be a nightmarish harbinger of ecological collapse. They are a keystone group for the aquatic food web, and when the keystone crumbles, so too does the larger structure. Alright, that's it for this episode, folks. I hope you thoroughly enjoyed this roller coaster of a show as we explored the extremes and horrors and the amazing parts of these smaller, more basal crustaceans. If you enjoyed the show, then hit that like button or give the show a 5-star rating. The next episode will introduce the decapods, and we'll begin exploring the larger crustaceans like the lobsters and the crabs. These future episodes should be just as wild as this one, so hit the subscribe button if you want to check them out right when I post them. If you want to support the show in other ways, check out the official store, or become a Patreon supporter with tiers as low as $1 and $2 a month. All of your financial contributions on Patreon and through the store, it keeps the lights on, it pays for recording equipment, it pays for hosting fees. It really goes a long way, and I deeply appreciate it. And as always, thank you for listening.